Hello, everyone. It's great to be here representing AlloVir, our team of 100, and to share our story. My name is Diana Brainard, and I'm the CEO of AlloVir, as well as an infectious diseases physician. I'm particularly excited today to tell you about what we're doing at AlloVir because of the strength of the scientific underpinnings of our technology and the clinical data generated to date, as well as the tremendous unmet medical need that immunocompromised patients have when facing these devastating viral illnesses with limited or no treatment options. And because of the highly experienced and dedicated team that has come together to make a difference for patients. At AlloVir, we are leading the field in developing allogeneic, off-the-shelf, virus-specific T-cell therapies to treat and prevent life-threatening viral diseases. We're approximately 1 year post-IPO, and as a public company, here are our disclaimers. Using virus-specific T-cells to treat immunodeficient patients with life-threatening viral diseases has been demonstrated and validated in multiple case series for more than 2 decades. This is an approach that's worked and being used right now at a small scale at multiple high-volume stem cell transplant centers throughout the US and globally. What AlloVir has done is take key advances in this technology that have come out of Baylor College of Medicine that enable this bespoke approach to be scaled to become a product. These key advances include, firstly, figuring out how to make a multi-virus product so you can target more than one virus at a time for treatment and prevention. The second is demonstrating that with these virus-specific T-cells or VSTs, you don't need full HLA matching for efficacy. This enables the creation of a bank of off-the-shelf allogeneic cells that can be created from a small donor pool. With this technology, we have created a rich pipeline of products that span the preclinical space all the way to phase 3. Now, more than ever, we can all appreciate the importance of continuing to invest in and develop antivirals. On the left is a list of 12 different viruses that we at AlloVir are currently targeting across our pipeline. Individually, some of these viruses are really very rare. However, collectively, there's a tremendous burden of illness, particularly among high-risk populations, for reactivation or primary infection from these pathogens. And these manifestations of disease cross all organ systems and can be truly devastating. Most of these viruses have no or inadequate treatment options, so there's significant unmet need. Our lead product is posoleucel, also known as AlloVir 105 or Viralim M, and it targets 6 different viruses that cause significant morbidity and mortality, particularly in high-risk hematopoietic stem cell transplant recipients. That's to say BK virus, cytomegalovirus, adenovirus, Epstein-Barr virus, human herpes virus 6, and JC virus. I want to highlight that we're in phase 3 right now with our lead indication in hemorrhagic cystitis, typically caused by BK virus and to a lesser extent adenovirus. We also have 2 critical proof of concept studies that are ongoing, one in the prevention of these 6 viral diseases and the second in kidney transplant recipients with BK viremia. We're on track to see data from both of these proof of concept studies later this year. In addition to posoleucel, we have a multi-respiratory virus product, AlloVir 106, that targets RSV, influenza, parainfluenza, and human metapneumovirus. As COVID restrictions have loosened, cases of these infections are starting to rise again, particularly in the South, and we'll start this trial by the end of the year. We've also initiated a single virus VST for SARS-CoV-2, which is targeting immunocompromised patients. Our approach targets epitopes on the virus that are not impacted by the viral evolution driven by antibodies, and we've seen strong potency persist as the virus has mutated and novel variants have emerged. Finally, I want to highlight our HBV program, AlloVir 107. We're completing our preclinical experiments this year to support moving into the clinic by the end of next year, and are really excited about the potential to redirect the immune system in this chronic viral infection affecting hundreds of millions of patients globally. Before sharing some data, I want to also highlight how the approach we're taking is unique with a broad range of intellectual property protection covering our innovation. Our product starts with the selection of targeted epitopes that confer immunity for each virus. This is a rigorous process that's been honed by Ann Lean, our Chief Scientific Officer. Healthy donors with immunity to the viruses are selected, and we have a proprietary algorithm that ensures that we cover the diversity of HLA types needed, not just for the US, but for the global population with a manageable donor pool. Cells are then expanded through a process that enables retention of broad spectrum multivirus activity, polyclonality, and a high degree of potency. The cells are then cryopreserved and have long-term stability. When a suitable recipient is identified, we have a patented matching process that can identify the best match for the patient, and we can get those cells to the patient within 48 hours. I now want to jump into our lead product, posoleucel, and share some of the exciting phase 2 data, which has given us so much confidence, and also where that's taken us in terms of our clinical priorities, focusing on transplant patients and working to relieve them and healthcare providers of the burden that these devastating viral infections bring. Posoleucel was studied in a phase 2 study we call the CHARMS study, run out of the Baylor College of Medicine. This proof of concept study looked at 59 patients who had refractory treatment-resistant viral infections due to one or more of the 6 viruses that posoleucel targets. Patients were treated and then followed for up to 12 months for efficacy and for safety. What's shown here are the dramatic results of that phase 2 open label study. There was an overall response rate of 93% of patients exhibiting either a partial or a complete response to treatment. 11 out of those patients experienced infections due to multiple viruses, and their response rate was 100%. This data was the primary underpinning for receiving RMAT and PRIME designation in the US and Europe, respectively, and has driven us to expand our phase 2 and phase 3 programs across a range of indications. Importantly, the study also showed that virus-specific T cells have a safety profile that's highly encouraging. The infusions were well tolerated. These cells are not gene edited and are not associated with cytokine release syndrome to date. There were also 14 cases of graft versus host disease, a complication that we carefully follow. 8 of those patients had pre-existing graft versus host disease, and 6 patients with de novo graft versus host disease were all grade 1 with skin manifestations that resolved with topical treatment. Importantly, these rates were not above what would be expected as a background rate in a hematopoietic stem cell population. Based on these data, we've worked with regulators in the US and globally to establish the design of a phase 3 registration study in virus-associated hemorrhagic cystitis. This is a double-blind, placebo-controlled trial in 125 patients who are randomized to receive either posoleucel 2 doses separated by 2 weeks or placebo. And the primary endpoint is time to resolution of hematuria. This study is important because we're targeting patients with the most severe disease, grade 3 and grade 4. And these patients suffer significant morbidity and even are at risk for kidney damage and transfusions. This study is actively enrolling, and we look forward to having results in 2023. Among hematopoietic stem cell transplant recipients who are high risk, the rate of infection or reactivation with at least one of these 6 viruses that posoleucel targets is nearly 90%. And what you can see here is nearly 2/3 of patients reactivate with 2 or more viruses. So there's a significant need to be thinking about how we can move further upstream and make sure that these patients don't suffer the consequences of these viral diseases. This is the underlying drive for our prevention proof of concept study. In this phase 2 study, which is the first of its kind, looking to prevent clinically significant disease across all of these viruses, high-risk allogeneic hematopoietic stem cell transplant patients are being randomized in the second part of the study to posoleucel or placebo. This part of the study will follow an open-label cohort that's currently enrolling, where we plan to look at 25 patients worth of data to understand the safety and early viral efficacy of trying to prevent viral disease from emerging in patients who either have no viremia or have low levels of viremia without signs of disease. We expect data from this open label portion of the study by the end of this year. I want to now shift gears and talk about another high-risk population, solid organ transplant recipients. These patients are also immunosuppressed, but in a different way from hematopoietic stem cell transplant recipients. In general, their immunosuppression is less intense, but more long-lasting. These patients can suffer significant consequences from viral diseases. On the left, you can see the graft survival rate among patients who develop the dreaded complication of BK virus nephropathy as compared to those without that complication. And you can see a clearly lower graft survival rate among those with BK virus nephropathy. Similarly, cytomegalovirus is a dreaded complication in all solid organ transplant recipients. Despite having options for both prevention and treatment of CMV post-transplant, we see resistance emerge, and we still see patients developing significant clinical illness, and more treatment options are necessary because CMV infection can lead to graft loss as well as death. To that end, we've started a proof of concept study of posoleucel in kidney transplant recipients with BK viremia. This study also has 2 parts. In the first part, we're randomizing patients with low-level BK viremia to receive posoleucel at one of 2 treatment intervals or placebo. As we generate data in this earlier stage disease, we'll then move to a group of patients with a higher viral load and pick one dosing interval to study further. This study is also ongoing, and we anticipate having early data from that first cohort later this year. Overall, the potential for posoleucel to help patients broadly who have immunodeficiencies of varying types is significant. If we start with the patients who are hematopoietic stem cell recipients who are suffering from these viral diseases, we think about potentially nearly 20,000 patients who could benefit. When you think about preventing disease rather than just treating the disease that develops, that number nearly doubles. And as you move into solid organ transplantation and beyond to other types of immunodeficiency, such as patients receiving chemotherapy, those receiving chronic immunosuppression for inflammatory disorders and other conditions, this number continues to grow. The potential for posoleucel and for our other therapies to impact patients' lives and help viral diseases is really tremendous. And so with that, I just want to end with a few key takeaways from AlloVir and our platform. We're really excited about having this ability to target viral diseases in a multivirus specific way and in a way that allows us to scale and make these treatments available globally in an off-the-shelf manner. At this time, we're targeting 12 different viral diseases with the potential to bring more under the umbrella over time. We have strong phase 2 data with a 93% response rate and have a vision of reaching more than 100,000 patients in the future. And we've got phase 2 and phase 3 studies underway to support and clinically validate our programs. In addition, we've got an excellent pipeline where we're bringing other products into the clinic over time, and we're expecting catalysts to read out in the years to come. Thanks very much.